evening, everyone. We are here at the second day of the Marine Diversity Education series. In the previous session, we have studied about the marine bacteria and marine archaea, and today we are going to study about the marine eukarya. We are having our educator Husna here. Husna, you can take over.
Thank you, Sanjeevan. So today we will be covering about Eukarya introduction and then more about Protista kingdom. So in the screen, I think you can see different pictures. So human or a man, a fungi, a plant. So all these living organisms, they belongs to domain Eukarya. So last week we covered about prokaryotic ones, which are archaea and bacteria. So today we will be covering archaea, the interaction. So next, yes. So here you can see, three main domains that we were studying about bacteria, archaea, and eukarya. Yes. Now, when it comes to characteristics of eukarya, actually, eukaryotic cells are larger than the prokaryotic cells that we studied last week. Okay, and also they have true nucleus, which is membrane bound. So last week we studied when it comes to prokaryotic cells, they don't have a membrane bound nucleus. So only the genetic material which is naked, so out there in the cytoplasm. So when it comes to eukaryotic cells, they have the true nuclei. So eukaryotic means it consists with true nucleus. So that's to be considered, and then they form single and also large complex organisms like human, as any other animals, they belong to eukarya. So they are very complex, we know. So like single organisms like protists, they also belong to the same domain. So we'll be covering that. So there are two types: single cell organisms, as well as very large complex organisms. And also they have membrane bound organelles. So last week you know that we studied about the structure of a prokaryotic cell. So in that we had different parts. So here like in eukaryotic cell, it's very different than the last one, because here we have some complex organelles. Like in prokaryotic, we had genetic material that is not membrane bound, some plasmids out there, and also the cytoplasm. So that's very simple form. But when it comes to eukarya, as they are forming complex large organisms, these cells are a bit adapted to do functions inside the cells. So to do that, they have different organelles which are membrane bound. Okay, mostly they have membrane bound organelles. So let me explain like this. Like imagine like you have a big a polythene bag. Inside that you're adding some semi-solid jelly-like thing, okay, and a plastic ball inside, and few different shapes objects into it, and you're tying that. So you can imagine a eukaryotic cell to be such model, because like you can imagine that plastic ball to be this nucleus, so and all the other small objects to be these organelles. So actually it's very complex inside the cell. So yes, so here you can see from the nucleus, there you can see some foldings are coming out, and that's attached to this cell membrane. The outer one is cell membrane, this is nuclear, yes, that's covering the nucleus, we call nucleus membrane. So here that folding is coming, so from the membrane they are getting folded and they are making this compartment like thing. So we call that endoreticulum. So the ones with small ribosomes on it, we call it's rough endo plasmic reticulum, while the one with no ribosomes, we call them smooth endoplasmic reticulum. So, and also we have here small we can imagine that to be noodles or something put into the that bag. Yes, imagine that model. Yes, so that can be said as these microtubules, or we have small microfilaments which support the cell. It's said kind of skeleton. So we know that our skeleton gives strength to our body. Such way, these microtubules in eukaryotic cells inside the cell, they provide kind of support, and they aid in movement, the changing their positions. So that's called there's microtubules. That's how they work there. And also especially we have mitochondria and also Golgi apparatus. These Golgi apparatus also some kind of folded compartments which is detached away from this nucleus. So I think this is a small introduction about eukaryotic cell and the structure. So like what I wanted y'all to work on this is like y'all can have this diagram, and you can to your project work, you can go home and then search about these organelles, different organelles. So they are doing different works. So when it comes to like mitochondria, so they are the powerhouse. Okay, yeah, they produce energy to the cells. So this is a brief explanation that I'm giving. So going home, what you can do is like y'all can like search what is Golgi apparatus, what is mitochondria. So you can have a nice sketch in your book, then write a small description what it is, and then you can have their functions as well. So this is a very basic thing so about eukaryotic cell. So next we have, yes, a comparison chart. So last week we studied about archaea and bacteria, the comparison between those two domains. Today I have attached the eukarya as well. So like when it comes to composition of cell wall, we are now confined to one particular thing. We are checking whether peptidoglycan is present or not. So when it comes to archaea and eukarya, the peptidoglycan is absent. So, and when it comes to bacteria, yes, they have peptidoglycan cell wall. Okay, type of cell membrane. So archaea and eukarya, they have lipid, bacteria and eukarya, they have lipid bilayers, but mostly archaea consists with lipid monolayer. Sometimes bilayers also, like generally we say monolayer. membrane lipids, archaea and bacteria. Sorry, yes, only these archaea consist with ether linked one, while bacteria and eukarya, they are ester linked. And when it comes to marine main tRNA, archaea and eukarya here, they have methionine. So bacteria, bacteria have formyl methionine here. So when it comes to number of DNA polymerase they have, so archaea and eukarya, they have multiple DNA polymerase. So when it comes to bacteria, they have single. So here in this chart, what we want to focus is like the characteristics that's more related to genetic material or the genetic of this organisms. Like you can see archaea and eukarya, they are having a bit similar thing, like main tRNA and number of DNA polymerase, that's more related to there's genetic material or the genetic study we can say. So they are close, they are very close. So that's why the scientists, they categorized archaea and eukarya like we studied in the last time in the polygenetic tree, bacteria taking from the ancient yes, the cell. Bacteria is taking a very prominent or distinct branch, and then from the another branch, bacteria and archaea, archaea and eukarya are coming out. So that means bacteria is very different from eukarya and archaea. Eukarya and archaea sharing kind of similar thing that's more related to their genetic materials. So that's what I wanted to highlight from this chart. Yes, so from the domain eukarya, this can be subdivided into 4 kingdoms. So we'll be studying, like it's a large domain. So when it comes to eukarya, all the human, the animals, fungi, plantae, all these things come under eukarya. So this can be subdivided into 4 main kingdoms. So we'll be covering about protista, plantae, animalia, and fungi one by one. So today we'll be focusing on protista. So I think before we move into the next in detail to study about protista in detail, we have one group activity prepared for y'all. So we hope to set y'all into breakout rooms. I think Sanjeevan, we can work on that.
No, I can divide them into groups, but you can elaborate about the questions and how they want to work on it.
Yes, so we will be putting you all into 2 breakout rooms, main 2 breakout rooms with time consent. So there we will be sharing you one activity. It's a very simple one. So you all can feel that. So why we give such breakout room session is like you all can get to know each other, the students who are taking part in this. So you all can make friends, and while discussing, so you all can have more idea. So one person can come up with one particular answer, so it can be either right or wrong. So that way you can discuss among yourself and come into a good understanding. So that's what we want. Yeah. So Sanjeevan, we can put them.
We'll be sharing the questions in our WhatsApp group. You can have a look at those
Yes,
questions.
yes,
And
that's true.
you will have 10 minutes time to complete all those questions, and then you will come back to this room. Yeah, you are being invited to join the breakout rooms. You can join your rooms.
Yes, so we are now going to study about Protista, kingdom Protista. So Protista, mostly they are aquatic and usually unicellular, or there can be a multicellular organism as well. So they can reproduce asexually and sexually. And when it comes to their feeding, they can be heterotrophic as well as some they belongs to autotrophs. And we know that autotrophs they can produce their own food. So these protists they are in marine oceanic food chain or food web uh, because of their the ability to make their own food. They serve as the base of the ocean food web. So here we can divide protist into three categories. So like the types, so animal-like, plant-like, and granule-like. So for our easiness to understand, so I have put them into three. So animal-like, plant-like, and granule-like. So we'll see one by one. So yes. So like animal-like means like they are called protozoa. So animal-like are called protozoa, meaning first animal. So because they started accumulating, like they started to take their food from outside, from the environment, and also they showed some movements. So yes, so with that they are known as first animal. So mostly they are unicellular and heterotrophic, so they don't make their own food, like they have to depend, and they prey on microorganisms. They engulf and digest. We call that phagocytosis. So they engulf the food and then digest. We'll be studying that in detail in the coming slides, so don't worry. So animal protists lack cell wall. They don't have a cell wall, so they can show free movements with the aid of microtubules and microfilaments inside the cell. So that's why we call them animal-like. So there are four main types actually we can study. One is amoeba, the other one is flagellates, which with a flagella, and then we have ciliates, yes, pseudopods. Yes, next. Yes, so here animal-like protists, they show some movement. Like earlier I said. So yes, they have a pseudopodia, which means a temporary like extension of this cytoplasm. So cytoplasm is from that model that I already described to you all, it's the semi-solid gel inside the bag. So you can adjust it, squeeze it to any direction that you want. So that's how these amoeba also work. So when they want to move, they just come out with an extension of their cytoplasm. So the body, they move forward. So that's called pseudopodia. Pseudo means not true. So podia means we can say foot or arms, so which is not true. It looks like arm or foot. So pseudo means that. So organism which exhibit amoeboid movement, we call this kind of movement amoeboid movement. Okay, so the very general example here we have is amoeba. So here you can see these are pseudopods. So time to time they can change the structure of the pseudopodia. Okay, and then you have, yes, similar, the other things are similar to eukarya cell. So some amoeba are predators. They show they want to consume, like they prey on, okay, they consume bacteria and other protists. And then when it comes to there are some other kinds, like they are detritivores, okay, they prey on some dead materials, dead organic matters. So we call them detritivores, okay. So like the ones who prey on the living organisms, small bacteria, and we can say other small protists. So they are predatory, okay. Yes. So when it comes to flagellated protozoans, so something here I have displayed a Euglena in this slide. So actually they are like in the first class I said about flagella. So here also this protists, they have a flagella to move. So in this case they don't have a pseudopodia, they don't form pseudopodia, they have their flagella. So they, with the help of flagella, they move, okay. So here you see Euglena. So this actually Euglena exhibit kind of animal-like and plant-like, because the reason is Euglena, they have like photosynthetic pigments in their body. So we can place them under plant-like. At the same time, we can say it's a kind of animal-like flagellated, okay. So they show movement, okay. So when it comes to this Euglena, they are single-cell flagellated eukaryotes, and then they are found in freshwater and salt water, okay. They are autotrophs, photosynthesis. Sometimes can act as heterotrophs. So I said because of the pigments in the body, they can do photosynthesis as well. So contain chlorophyll A and B. So just like plants, contains minerals. Okay, they are very rich in mineral. So they are used in food. Okay, they can be taken as a food source from marine environment. So next we have dinoflagellates. So earlier one, they had one flagella, okay. Here we have two flagella. So they have two flagella. So these kind of small creatures, they have two parts. That means upper and lower tests. Here you can see clearly upper one and the lower. We call them tests, or these tests are rigid, okay. They are very thick. They protect the animal inside, okay, the organism inside. So tests are made up of plates, and this contains cellulose encrusted with silica. So these are like they are made up of small plates, so that's with cellulose encrusted with silica. So combination of cellulose and silica, okay. And when it comes like they're feeding, they can be symbiotropic, okay. Last time we studied about symbiosis, okay. So that means they can live together, okay. Symbiotropic means they can obtain their food by living together. Like they have another type, they can be as epibiotics as well. So epibiotic means they can be attached to one surface of animal or plant. So epi means they live on a surface of a plant or an animal, so that way they can get their food. So these are dinoflagellates mostly, yes. So next we have some examples to go. Yes. So here you can see this is a first example is Dynamostigota. Okay. So they are very minute organisms. So they are abundant in warm seas, produce powerful toxins, contribute for red tides. So here in the upper part you can see something very unusual color change in the ocean. So those are called red tides. Okay, red tides they can appear suddenly. So due to this small organisms, they come like they bloom at once in the ocean. So they give color. Okay, uh, because of that, the color of the ocean changes. So it's very unique color, something that's related to red, a pink, okay, yellowish, orange. So we call that red tides. Okay, so these are very kind of harmful ones because these organisms, they produce toxins. So when the fish or any other organisms, when they consume these, when they come into contact with this uh, red tide and these organisms produce toxins in the environment, it can be harmful. And also like we sometimes can develop some allergies to that. So that's all about red tides. So for your project, actually, I want you all to read more on red tides. What are red tides and how it's happening, and what are the impacts for human as well as other organisms in marine. So you can like you can do a good description about red tides, I think. So keep that in mind. And so next, our next example is Noctilucer. So Noctiluca, okay, and they appear as twinkling light in the sea during night. So it's a very interesting phenomena. So in the next slide, you can see that. So here, so they are the creatures. So here you can see. So this is how the sea appears during night because of their bloom. So they are abundant in sea everywhere. So when wave comes and go, this is how it appears. Okay, so this is called bioluminescent. So which means due to chemical reactions inside this Noctilucer body, they produce light. Okay, so this is very interesting. So you can search about this as well. So next is about Serotiem. So Serotiem, there are the several are known to cause red tides as Dynamostigota. So yes, here there's we can say they cause red tides and water blooms. Okay, the cell contains chromatophores, which means they have pigments. Okay, colors. So yellow, brown, green pigments. Okay, especially for Serotiem. So common in fresh water and also salt water from Arctic to tropic. So we can have Serotiem in all like all the oceans. So from Arctic to tropics, it's common. Okay, so have both animal and plant-like behavior they show. Okay, so the next is about Gymnodium. So this is a kind of a dinoflagellate which is present in marine and freshwater. Okay, and also they also exhibit animal-like and plant-like characteristics because they also have these pigments. Okay, and then when it comes to this, some may be bioluminescent or form a periodic water blooms that may occur water yellow or red. So they also contribute for these water blooms, which especially yellow or red, changes the color into yellow or red. Okay, and also they produce toxins, okay, similar to that of dinoflagellates called goniacs, correct? So both toxins are fatal to which is harmful to fish and can irritate the nose and throat of humans if inhaled. So if the bloom is there, like if we go if we walk in the beach, so like if we inhale the air, so it's gonna irritate our nose and throat due to their toxins. So it's very like we have to be more careful, like while we have this blooms. Okay, and then yes, some species are photosynthetic and others are heterotrophs. So that's clear, I think, because they have the color pigments, so they can do photosynthesis. So the other next example is zooxanthellae. So this is something interesting. So in the picture you can see to my left I have a very colorful picture. So can anyone tell me what okay, or already its description is in the slide. Anyway, this is about a coral. So in the left you can see very colorful coral, and to the right yes, you have a very white color which is pale looking, so no color. So the reason is like this is a live coral, so that which means they harbor or they have zooxanthellae in their body. Coral polyps, they hold zooxanthellae. So zooxanthellae are in a symbiotic interaction with these coral polyps, which means living together. So zooxanthellae is a dinoflagellate. So they are in a living together form. So they provide coral with pigmentation. They give colorful, like colorful colors. Okay, we can say pigments. So on the left, there's a like healthy stony coral here, and on the right is a stony coral that has lost its zooxanthellae. So whenever you see a coral without color, you should know that they have expelled, they have sent out their living together partner, zooxanthellae. So that's why they lack the color. There, you understand what I say, okay? So that's how it appears. So when they expel the zooxanthellae, they don't have their own color corals. So we should know that. So if coral polyp is without zooxanthellae cells for a longer period of time, it will most likely die. Yes, this happens. So one can ask why they expel zooxanthellae like while being there. So this happens due to the oceanic condition changes. Like for corals, they have to have a good temperature, okay, to survive. They need temperature which is 27 degrees Celsius to 30 degrees Celsius, something very, there's a range, and then they have to have very clear water without turbidity. So whenever this condition changes, when a heat wave or when the ocean temperature increases, so they expel these zooxanthellae out. So they become this like very pale. So when if the condition is very temporary, so if seas cool down, so again they can harbor, they can come and start living. So Sorry for that. Yes, so they can start a living. So if that condition prevails for a long time, they go die. So that's all about zooxanthellae and corals. So in the next slide, you can clearly see how here see like this is the coral polyp. So these zooxanthellae, they provide photosynthesis. So through photosynthesis, they give nutrients to the polyp, okay? And the same way, they give oxygen as well. So in return, these coral polyps, they give shelter. So they give a protected place to live, and then they give carbon dioxide to do photosynthesis. So that's the interaction between zooxanthellae and coral polyps. So here, a temporary state, we can say bleaching, and then if it's the prolonged condition, with the prolonged condition, they can go die. So we call corals are dead. Okay, so the next, it's about ciliates. So yes, we saw about the flagella, okay, which is very long. We can have one or two, but when it comes to cilia, they are similar structures, but they are short. The length is short. So all over the body, cilia can be there. So it depends. It depends on the organism. So we'll see. Okay, the ciliates live in freshwater and marine environment, and they're short. Okay, all are phagotrophic, consume bacteria. Single cell organisms, free living, or they can be parasitic. So we have here 4 examples. We'll study one by one. Okay, here we have Paramecium. So Paramecium, here you can see these short structures, we call them cilia. It appears to be throughout the body, all over the body. So the specialty is like if they find small organisms, whether it could be bacteria or any other small protist. So what they do is using this cilia, the movement, they gather the food particles towards their oral groove. See here you have this oral groove, which is mouth. So they aid with their movement. They facilitate the prey to be in the oral groove, and then when they go inside the oral groove and buccal of overture, so inside when they go into the body, they form small bags like thing. Okay, so once they form the food vacuoles, bag like thing, so they can be circulated throughout their cytoplasm. So while they move here and there, they get all the enzymes. Okay, so we know that like in our body, if we eat, it will go straightly into the stomach, and we have all the digestive enzymes working there. So we know the process, but here it's very different. So they form small bag like thing, food vacuoles. So those like they can be transported inside the cytoplasm. While they move here and there, they get digested. So once the food is digested and the nutrients are absorbed to the cytoplasm, so through anal pore, they are expelled out. So here you can see they are taking in, making bags. So it's getting digested. Finally, it's out. Okay. So yes, so they can be found in freshwater, brackish water, and marine water. Okay. So that's all about. So they move using this cilia. So the next is Didinum. So this is you can see they are barrel-like structures. So in two bands, they have cilia, not all over the body. But here in this light blue color, you have paramecium. So all over the body, you have cilia. But when it comes to Didinum, it's very confined to two band structure. So you have cilia here. So yeah, this specialty about this Didinum is they consume, they majorly prey on Paramecium. So they can consume other protists or bacteria as well, but this is the specialty. So here you can see how it's taking the Paramecium in. So after taking the Paramecium in, they appear very big barrels. So that's Didinum. So it's very interesting. So you can see you can watch videos out there in YouTube, so how they feed on this paramecium. So the next is Acineta. So this is a free living but a sessile one. So they are confined to one place. So you can see they have a chalk-like thing. Yeah. So that's how, like here you can see that's how they get attached to one surface. So usually to any marine organisms, they get attached to the surface. So in two sides they have some elongations you can see. So those are called feeding tentacles. Okay, so these feeding tentacles are always here like this. So they always look for prey. So once when they get a microorganism or bacteria, in the tentacles, they have one special organelle called hepatocyst. So through that, they paralyze the prey. So when the prey comes, they close it, paralyzing inside the body. So that's inside the body, food vacuum. So they form. So that's how it works. Okay. Yes. Next. So granuli. So here it's all about forams. So forams are very important when it comes to marine protists. So here in this picture, you can see that they have studded pores in their body. That's the shell. Okay. So in the shell, you can see small pores. So what happens is, this is the shell. Inside there's cytoplasm, the jelly-like thing. So through this studs, this pores, actually the cytoplasm comes out. So when the cytoplasm comes out, they appear like thin filaments. So here in the down picture, you can see very nice elongations all over the body. So that's yeah, that's the cytoplasm, how it comes out. Okay, so there's thin filaments. Okay, these thin filaments, they are used for swimming, feeding, and collecting materials. Okay, exclusively marine. Smallest ones are around 10 micrometers, but there are large ones we can see through our naked eyes. Okay, so that's all about. And also they have brilliant colors. Okay, varying colors. So the one example I can state here is Globicarina, so which is a free swimming one, planktonic, which floats in the column. So, and they are food for marine mammals. So marine mammals, they eat on it. And when they die, okay, actually when these forams die, they go slowly and then they settle in the marine sediments, okay, marine flow. So ocean flow. So they form with mud, they form a layer consisting of this shell material. Okay, so those are called ooze. Okay, especially this word ooze. So they are sedimentary layers consisting with these shell particles. So layers in marine sediments. Okay, next is about Radiolarians. Okay, so when it comes to Radiolarians, they are very spherical in shape, and they also have these extensions from the body, but there are two types. So here I have mentioned Exopods and Pseudopodia. So there are two types of elongations of the cytoplasm. So here in this picture you can see some are very out, they are going far from the body surface, some are very close. Okay, so the elongated ones are called exopods, while the small extensions are called Pseudopodia. So okay, so yes, like they radiate from the body. Okay, and exopods found to be useful in catching prey, and they also help in rolling. Okay, so in movement. So and also we have to consider this exopodia, they can be elongated, I mean they can be adjusted, they can be shortened, or they can be elongated according to their preferences in the movement. Okay, and then here we have some examples, Acanthometra and Polycystina here. So you can see how their shells appear. So I wanted to highlight the shell structure. So yes, and then we have our next one here. This is what I wanted to highlight. So it's very spherical in shape, most of them. So radiate from all like they in a what we can say in a general, like in a pattern, the cytoplasm radiate from the body. So that's why we call radiolarians. So yes, so when it comes to plant-like protista, so plant-like protista, they are photosynthetic. So we usually call them algae. So and also we can address them as seaweeds as well. So they have different types of cell walls. So some people, they confuse these algae or seaweed to be a true plant, but actually they are not true plants because they lack leaves, stems and roots. So to be a true plant, they need to have root system, stem and leaves, but they appear very green, large. Actually, yes, sometimes people get confused with that appearance, but they are not true plants. Okay, so reproduce asexually or asexually. Most algal species are unicellular or multicellular. Okay, live in aquatic environments, ocean and lakes. We can have they are the freshwater as well. Okay, so here I have put it everything in one chart, so it's very easy. This long I was giving all the pictures and small descriptions, so this gonna be like you can have a look at it at once. Okay, so in this chart, so when it comes to red algae, so cell wall composition is polysaccharide. So chlorophyll pigment is phycobilin. So when we consider about the environment, it's marine, mostly marine. And when we we know that they do photosynthesis, so food type means the one like the storage, the food storage is called fluoridine starch, and mostly they are multicellular. Example, Gymnodium. Okay, so when it comes to green algae, their cell wall is composed of cellulose, and they have chlorophyll A and B. So they are mostly terrestrial, marine, and we can have them in wet soil as well. So the food type they store is starch, and they can be multicellular and unicellular as well. So example, ulva. So when it comes to brown algae, their cell wall composition is polysaccharides, alginides, and their chlorophyll is, we can say, fucoxanthin. And the environment they occupy is mostly marine. So marine, and also they can be found, one type, this kelp forest, they are found only in the polar regions, okay, very cold environment, they are the ones there. So food type laminarin, and then they are multicellular, not no unicellular ones. So example, kelp forest and sargassum. So when it comes to yes, golden brown algae, we have the cell wall composition is silica, and then we have chlorophyll A and C, and then carotene there, and mostly they are marine. They can also be in environmental, like waterways and soil. And their food type, what the type they store in their body is chrysolaminarin, and they're unicellular, example diatoms. Okay, in the next slide I can show you some pictures. So these are red algae. So like when some person sees, they can confuse, yeah, they can mistake this with the plants, but they are red algae, so they appear like this, but these are not actual branch or leaves. Okay, so this is green algae. So this is ulva. So it appears like leaves, very big leaves, and then here you have small grapes, like sea grapes, so grapes like structures. So you might have seen sometimes in the ocean. Yes, here you have brown algae, so sargassum. So here you have buds, so they always keep on floating. So in the ocean column, so here down is the kelp forest. So the next topic that you should focus is kelp forest. So you're going to write about, like, you're going to search about what are kelp forests and how they function. So yes, you can have a good description. So I can give a short brief like an explanation to this. So actually this they are brown algae, so they can grow into big length, so meters and meters they can cover. And actually they can capture more carbon dioxide. So this is very confined to polar regions. So they are the ones, like here we talk about mangroves in tropics. So they are the heroes in polar regions. Okay, so here in the tropics and subtropic, we tell that carbon dioxide, like they take more carbon dioxide, mangroves, so we have to safeguard them the same way. When it comes to polar regions, they are the ones who is doing great job. So this is our sea otter. So in this, when it comes to this kelp forest, they take the main role there. So they are keystone species, we can say, and then they come into interact with sea urchin there. So yes, so once the sea urchin, yeah, when like, yeah, I'm not going into that deep. So yeah, there's an interaction between sea urchin to sea otter and then to the seagrass. So you can search, okay? So you're going to do a good picture, like you can draw a good picture about this kelp forest and then describe, okay? Yes, and then we have this golden brown algae. They represent different colors, especially yellowish, brownish, something like that. So muddy colors, especially. So they take different shapes, very small. They are very interesting. So small structures. So that's all about Eukarya and Protista, I think. So thank you for listening all this time. So yeah, thank you.